So one of the great things about coming into JP Morgan is we get to interact with a lot of our colleagues, especially as we go into a new decade. So I'm Tim Miller, and I'd like to have all of the panelists introduce themselves. We'll start with Tim down at the end.
Hi there. My name is Tim Liu. I'm the CEO and co-founder at Senti Bio. We're a synthetic biology company focused on programming genetic circuits into cell and gene therapy products. Thanks.
Hi, my name is Emile Nuwaysir. I'm the president and CEO of BlueRock Therapeutics. We are a platform company at the intersection of the cell and the gene. We believe industrial control and commercial rights to edit or make any cell and edit any gene in our therapeutic areas.
Hi, I'm Shelia Mikhail, and I'm the CEO and co-founder of AskBio. AskBio has been around since 2001. It's based on the work of Jude Smolsky. A lot of people say, oh, I don't know of AskBio, but they do know of some of the companies that we spun out. Bamboo Therapeutics, which was working on Duchenne's muscular dystrophy, was sold to Pfizer in 2016. And Chatham Therapeutics, which was working on hemophilia, was sold to Baxter, now Takeda, in 2014. Today, we're known for our Pro-10 manufacturing cell line. It is the highest yielding cell line in the industry. We sell capacity through the Virogen brand name, and many of you are our customers at Virogen, so thank you for that. We recently backward integrated into plasmid production in a joint venture with Touchlight Holdings. We now run Touchlight AAV. We are in the clinic today with Pompe and heart failure.
Hi, thank you. Laurence Cooper, CEO of Ziopharm. We're working on non-viral gene transfer to target solid tumors and also cytokine biology driven with IL-12.
Thank you. So let's set the stage for just a moment. It was 1945, and over 500 delegates from the United Nations came to celebrate and honor the recently deceased Franklin Roosevelt, and they went to Muir Woods. And if you have an opportunity to spend an hour or two there, you're struck by how the things that we in pharma and biotech, what we build, lasts for a long time. So as we look over the past 10 years, and we look at 2019 and look into 2020, the market has continued to throw a lot of money at gene therapies and cell therapies. Since 2015, as Jen mentioned, there's been close to $40 billion in funding in the space, and the market's IPOs has dramatically increased, with companies ranging an average of $200 million. But most of these therapies that are on the market are indicated for small patient populations, and how are we going to think about how they grow into larger populations? So I think we're going to start today's panel a little bit with kind of the concept of data today, cure tomorrow. And I asked some of our panelists about how they think about the next generation of AAV cell therapies or cell therapies. And maybe Sheila and Tim, maybe you can kind of kick us off with maybe a case study that you think about when you look back on 2019 and going into 2020, what we might think about from a new manufacturing process, a new AAV. These are the things that are going to help transform our space. Sheila, you want to kind of start?
Sure. So we've always had to think big. One of our lead indications from the beginning was Duchenne's muscular dystrophy. AskBio was started as a collaboration between researchers and parents who had children with devastating diseases. And one of our founders was a parent who had a child with Duchenne's muscular dystrophy. Now, Duchenne's is a very complicated disease, but more so, it's complicated to make enough material for it. And so that was one of the big issues that we had to face from the very beginning when we started the company. So we started off thinking, how are we going to manufacture in large scale? And in fact, we were very hesitant to bring our therapeutic into the clinic until such time that we resolved that problem, because we knew that the Duchenne's community would be very upset with us if we could provide treatments and potentially cures to their children, but yet we could only give those treatment opportunities to a few patients. And so we started from the very beginning thinking about how to make sure that we could manufacture enough. And our Pro-10 now is the highest yielding cell line in the industry with 10 to the 17, and it is a very good quality material that we produce with few empty particles. But that being said, we're now working on things like heart failure. And heart failure is another really big market opportunity. So we have to think even bigger yet. So we recently purchased a company called Synpronics, and I see my colleague David Venerables, the CEO of Synpronics, in the audience. Sempronics has a really nifty inducible promoter, and that inducible promoter will let us develop a stable packaged cell line, which we're working on very rigorously. And so we keep thinking about the future because we've been here since 2001. We idled for a long time when there was no money in the industry, and we want to bring our therapeutics to as many patients as quickly as possible. Manufacturing is key. And I would just want to highlight one other thing. Plasmid. Plasmid is a big rate-limiting step for a lot of us. Aldebaran is a great partner to each of us, but they are capacity constrained, and we wait a long time for our material. It's a big rate-limiting step. And so we backward integrated into doggy bone DNA, which is a different way of making the starting material. It is faster because it's made in a test tube with a bioenzymatic process rather than E. coli with big bioreactors. It is cheaper and it is safer, we believe, because it has fewer bacterial contaminants. So manufacturing is the key, but it's just not the virus manufacturing, it's the whole supply chain.
Thanks. Manufacturing will certainly be a big topic for later on in the panel. Tim, do you have some thoughts?
Yeah, I think, well, if you think back to the last decade, it was very exciting times. We started at the beginning of the decade without that much excitement and interest in the cell and gene therapy space, and we're obviously ending it, but we have ended it with tremendous amount of excitement and interest. And I think one of the key things that, looking back, was really impactful over the last few years is we have clear evidence now that, genome modification, whether it's through introducing a gene or modifying a gene, can be safe, can be efficacious, and really be transformative for patients in specific indications. And so for us, I think thinking about what that allows us to do is that we can start really opening up the way we think about therapeutic problems and the aperture at which we try to tackle some of these diseases. We can ultimately think about really changing the genetics of the disease rather than simply just trying to modify with a small molecule or biologic. And so I think that's fundamentally one of the big things that's happened over the last decade. It really has opened the mind, and really we see it as an output of all the work that has gone into DNA synthesis and sequencing to get us to this sort of point. I think answering to try to address some of the questions that Tim brought up earlier, what's the future going to hold? I think we need to try to figure out how to broaden and enable greater access of cell and gene therapy into other indications. So whether that's reducing cost of goods through allogeneic therapies or reducing the cost of manufacturing, as I'm sure we'll discuss, but also to try to address the sophistication of these therapies and how to get there, right? So how do we go beyond making single changes, single modifications, constitutive expression, which I think are inherently limited to certain types of diseases that can tolerate that, to ones that sort of enable greater control, regulation, targeting, and even multifactorial approaches. So I think it's pretty clear now from the basic research side that that's possible. The design cycle for modifying and making these sort of therapies is only going to accelerate over the next decade. And it's a matter of how do we then take that, pattern match that with the right indications and really drive those into the clinic.
Yeah, thank you. So I look at the decade. We started with proof of principle. The seminal work, especially in CAR-T with CD19, really established that genetically engineered cells could have therapeutic potential. We kind of got to the middle of the decade, and there really started to become a question of scalability. I think everybody's talking about that. Whether these programs would be essentially hit the mainstream, or would they be seen as salvage therapies? So, and I reflect on that a lot because I'm trained as a bone marrow transplant physician, and bone marrow transplant remains as a sort of an academic salvage-based therapy, very high cost, very small numbers of patients. And it is in some ways the preeminent example of cell therapy. So as we get into the new decade, that question of scalability, I think, is going to be really front and center. And if we don't scale, then it will become essentially either salvage therapy for high volume diseases, or it'll be very targeted, for instance, for Duchenne's and such, where children who have these devastating illnesses, and you can match essentially their quality of life with the high cost. So for myself, the scale part is really what drives my thinking. And I like thinking of ideas in which you can essentially take these foundational principles and then grow that technology. And so perhaps we'll get an opportunity to talk about it. Mm-hmm.
Emile, first thoughts?
Yeah. So BlueRock was founded on a slightly different principle, which was that there are certain diseases for which today, at least, the approaches that we've developed really won't work well. Diseases like heart failure, where after a heart attack, that patient loses 1 billion heart muscle cells. And so it really doesn't matter what small molecule, what large molecule, what device you develop, it is very difficult to replicate contractile force with a drug. And the thesis we had was that the only way to replicate that is with the native machinery, the cell itself. And so we believe that there was promise in the idea of a regenerative medicine where you could effectively reverse the degenerative condition by putting the cells back in. That's a big concept. That's why we started with a very large capital investment from a big partner. And, and we're going after diseases like heart failure and Parkinson's. Those two alone, there's 42 million people. And the standard of care for Parkinson's, while it's evolved a lot, is from the 1950s. So it's because it's a degenerative condition where once those cells are lost, it's very difficult to intervene. We're on the cusp of, we believe, transplanting healthy neurons back into the human brain and re-innervating the brain. So it's a different approach, and it actually, we didn't coordinate this, but your analogy to the redwoods and to the difference between transience and something that's stable, that's fundamentally what we do that's different, is that our goal is to manufacture and stably engraft the cell. We're going to stably engraft a manufactured cell, which has not been done before. And when you take that as your goal and imagine all the things that you have to do that, the manufacturing challenge to take that analogy is immense, because if you imagine 1 billion heart muscle cells is the dose for a heart failure patient, and there are tens of millions of people, the numbers are staggering. So, but I think it's absolutely possible. And if your question was in 2020, I think we'll see for the first time now we have a trial that's approved at the NIH. The first IPS, it's an autologous trial for the replacement of retinal pigmented epithelial cells, but Kapil-Bharti, and that's a step towards the theory of regenerating the retina. Hopefully our trial with replacing neurons. So there's going to be a whole series of these new strategies to do what is in effect the promise of 1998 with the first stem cell. We finally get into the point where we can make the authentic cell and maybe make a difference. So
No, that's great. Thank you for your initial thoughts. So, as we think about looking forward, right, into 2020, Shelia, so AskBio, Virogen, you're a big supplier now to many of the companies in this space, particularly from an AAV, standpoint. What do you think is going to be one of the key technologies or the key things you're going to be bringing, or that you're going to see in the field going into 2020?
I believe it's the plasmid. Again, that's the biggest rate-limiting step for production. It takes about 8 months to wait to get your plasmid supply, so that substantially delays the progression into the clinic. Also, it's a big cost of production. It's about 40% of your manufacturing cost. And so we have, again, with Touchlight Holdings, formed a joint venture for an alternative. We believe that the doggy bone DNA approach has a lot of benefits. It's faster, it's potentially safer because it doesn't use E. coli, and it has fewer bacterial contaminants. But we think it's the supply chain. I think there will be a few of us who will figure out how to do stable package cell lines like CHO cell lines. And again, we're all preparing for a day when we can move from the monogenic diseases and make our technologies available for patients in Parkinson's, Alzheimer's. The potential is there. The science will work. It's just a matter of how do we drop the cost of goods.
Yeah, big fan that the science always wins. Right, so certainly looking forward. We'll talk a little bit about regulatory interactions. I'd love to hear a little bit more about how some of the Touchlight and some of the regulatory interactions. I think hopefully the first IND might come in 2020. Emile, so we talked briefly about some engineering our cells, and how IO is really starting to become one of the really hot topics. Right. You want to talk a little bit about that?
Sure. So I think we would all agree that we've this profound advance of the engineered cell. It's changed the way we think about disease treatment, cancer treatment. It's proven that it's sort of, I would say, our generation's Jonas Salk moment, right? Something transformative that we invented. But it's also all the limitations in the manufacture, the stable engineering, all the challenges the previous panel is addressing or working on. But I think what I'm most excited about in the field for engineered cell therapy is maybe an example that was published at ASH just a few months ago by one of our peers, Fate Therapeutics. Shout out to them if they're in the audience, where they showed that you could use a pluripotent master cell bank to make the authentic cell, that you could make an NK cell that was of equivalent functionality to a cell that you could harvest from the body. And I think while that's just only one example, and they have multiple examples with serial engineering the master cell bank, it's profound because I think it harkens the end of a harvested cell therapy. I know that's like, we're talking decades, but until we've really fully explored the idea of a pluripotent master cell bank. But if you can make an NK cell that in vivo, in a human being, works just as well as a harvested cell, or better, I think there's something that's a sea change in the industry, in the paradigm for how to make a cell therapy. So that's what I'm personally, and I feel, to be honest, personally gratified because I've been on the lunatic fringe of this pluripotent cell movement, and now I feel like it, I was hoping that we would be the first to show that with our Parkinson's therapy. They beat us to it, but I'm cheering for everybody, so.
That's right.
Laurence?
So I don't think there's a one-size-fits-all solution. Base principles, when you're going after a cancer target, there's two broad categories of immunotherapies. The first category is the most advanced, at least with respect to clinical practice, and that's in the engineered CAR T. And in that situation, you have the poster child CD19, and perhaps BCMA will follow soon, where there is a homogeneous population of cells and there's one receptor. So in your mind's eye, you can go, okay, there's kind of there's just a library of one, and now essentially I have to get the library of one into everybody who has, for instance, the CD19 malignancy. So in that situation, the cost of goods, the scalability, then collapses to one of two questions. The first question is, will you use autologous cells? Because if you can, you may not have to lymphodeplete the patient. You can essentially get rid of a lot of the heavier engineering, but of course you've got a supply chain issue versus allogeneic. Both of those work when you have one receptor on the shelf. If you now turn over the page and you look at solid tumors, there, for instance, you can either go to CAR T or you can go to TCR-T. But in either situation, you need a complexity of receptors to match the heterogeneity of the tumor microenvironment. So there's two levels of heterogeneity. Within the tumor, lots of little pieces of the tumor need to be targeted. And then between the patients, lots of different types of tumors. So in that situation, it's harder to ascribe an off-the-shelf third-party cell because basically you've still got many, many, many receptors that need to be essentially put into those cells. So in that situation, you're probably going to an autologous approach, and you're probably then going to an autologous approach in which you can scale to hit all of those receptors. Because an allogeneic approach in which you have one cell line, you spend a lot of money, and yes, you can pre-make it, doesn't really solve the problem because you've got to put in all of those different receptors, and you're not going to make dozens, hundreds of different allogeneic cell lines. So I think, as we look at this, each piece of the puzzle will come into focus, but at the end of the day, solve the solution you're going after. Yeah.
All right, we're going to jump now into some more hot topics and talk about regulatory interactions as we think about the FDA and the EMA. Tim, I'm going to kind of start down there with you. So as we think about, there seems to be, I think it's well known, that there's a lot of turnover right now at the FDA, and there seems to be sometimes, as I think many of us have had interactions over the years, sometimes there's this sense that we always want to try and help bring our colleagues along. What's your sense going into 2020 when we think about how regulatory interactions are going to go?
Yeah, I think this ties in also where the frontier of the technology. I think all of us on this panel here and others in the field are really trying to innovate and bring new concepts to the FDA all the time. I think one of the things that we see going forward is a lot of what's going to go into cell and gene therapies is this idea of like a multifunctional program. So a cell that needs to do one thing and another, or an AAV that needs to, that's a novel capsid, but also can be controlled in some way. And so that's ultimately going to pose new, really fundamental questions to how we think about regulation. A lot of these products are a fairly new concept to folks. For us, so far, in terms of our interactions with the FDA, we had a pretty positive pre-IND meeting a couple months ago on our lead program. I found that the FDA continues to be very supportive and very thoughtful in terms of interactions with us. As long as you bring your plan and your approach to them with enough scientific basis to it. So what we tried to do early on was really lay out why we're doing things a certain way, do the sort of key experiments that any reasonable scientist would ask when you try to introduce a new technology. And so far, I think the feedback has been really positive. So I hope that trend continues. I think it will. And it's incumbent on us as from the industry side to continue to help educate and provide that sort of information, so we're not trying to bring things to the clinic that are too quick or take risks that would cast a pall on the entire field.
I think I've had the benefit of listening to Peter Marks talk about his challenges and opportunities and what he's recently and a few other people at the FDA. And I think what we're seeing is, well, first, our experience is that they're incredibly helpful. It has been an incredibly helpful interaction while they're doing their job, which is protecting the patient and develop it right under the appropriate control. So I would say that our experiences are they really are trying to be a partner to the industry, but I think they're a victim of their own success in a way. Some of the improvements we've seen, some of the dramatic acceleration, and the number of applications we're looking at, a challenge for the FDA because the number of applications is accelerating non-linearly, and the complexity of these applications, they're cells that are engineered multiple times, multiple engineering, multiple strategies, platforms. So I think we, as an industry, do have a challenge, which is how we can help the FDA to learn, because and of course we're all looking for experienced people that have FDA experience. So there's like this, perverse incentive for the people that
You're
get
looking
trained
for a job.
Yeah, and so I think we as an industry do have a challenge that we have to try and solve, which is how to help the FDA get up the curve on all these very complicated issues where we're effectively the only expert, in our area. I think I would point to some initiatives, for example, in my Small Royalty Alliance for Regenerative Medicine, we're patterning our efforts around manufacturing standards and best practices and approaches around the initial document, which was AMAB, which was sort of the standards for the manufacturing industry for antibodies. There will be documents coming on cell and on gene, a gene and a cell. And so I'd look to you to help us to improve that as we go forward and to contribute to it if you're interested. So.
Yeah, no, I think that's really important. I'm glad that that work is being done by ARM. I think right now one of the big issues in the gene therapy area is titering and knowing the comparability between different production systems and, are we really giving the same doses or is there some variance because of the way we're measuring our potency? So that will be important work, and I think it's something that the FDA is also looking for us to take the leadership on. I think the theme that I keep hearing over and over again is the FDA, and we have found this too, they're very collaborative and supportive, but sometimes they need to be educated, especially as we make new innovations. It's our responsibility to tell them why those innovations are important for patients and how they improve the patient experience, either in terms of safety or the effectiveness of our drugs. So it's really on us to come with scientific data and to explain our positions. And my experience has been when we do that, we usually get pretty good reception. I also think Peter Marks is doing a great job in trying to be innovative and trying to figure out how to streamline the processes so we can bring our treatments to patients more cost effectively and quicker. A lot of patients in the rare disease, especially where there are no treatment options, they have very devastating outcomes. And so it's a responsibility of the agency and for us to accelerate the approval process so we can get therapeutics that are beneficial to those patients quickly.
Yeah. So I think as I think about the regulatory process, I think in terms of how you can best partner with the FDA, there are really, I think, 4 major streams of activity that we can take part in and do take part in. The first is to de-risk the clinical enterprise by investigator-initiated trials. Often, lead academic centers with lead physicians have relationships with the FDA that goes back decades of trust, and that can be a very useful filter for the purposes of explaining biology and getting initial clinical data. I think the CIND process actually buds off that quite well. I think all of us have had patient experiences where the phone rings and a patient really wants access, and that can be very, very helpful for not only the patient but also I think for understanding N of 1 biology. The sort of those two, and then the third is looking outside of the United States for the purposes of obtaining initial clinical data. Struck by Greater China. And their regulatory processes that are going on, the way that they're able to organize their, essentially their approval process in a local regional environment, particularly tied to a hospital. There is, a great opportunity to essentially have a win-win biology, so you can learn. And then I think the fourth way to help the U.S. regulators, of course, is with the company-sponsored IND. And to me, that looks like the kind of the last step in the process, so that everybody's on the same page, everybody knows what's going on. There's really a wealth of data that everybody can then react to. Mm-hmm.
So there are over 1,000 active clinical trials. And one of the talking points that I think many of us have used in some of our decks has been 10 to 20 approvals by 2025. How do you think that that's going to be reflected in 2020? And I'll look to you, Laurence, to start.
So I think we have to kind of pause for a second. So I think the one way to think about it is sort of the buckets. So CAR T, so thinking about oncology, there I think there is real momentum. There's a playbook now for understanding, how to get other CARs approved, particularly for hematologic malignancies. I think as companies come essentially through their registration process, that to me looks fairly formulaic. There's essentially the set of rules. Where there's going to be great pressure is whether those approved products will make money. So actually that will be a real sticking point. So I don't necessarily, at the moment, go in the CAR T space approval, profit, success. I kind of go approval, and then I put a bit of a bracket around it. The sort of outside of the CAR T space where there could be real, essentially, momentum will be in, I think, for my world, in the oncology world, in two big camps. The first will be in TCRs that are now genetically modified to go after tumor. There, I think we're going to see some real durable responses, some real sense of momentum there. And that, I think, will kick off towards the rest of essentially the decade, approval processes for the TCR space. And that looks really pretty fertile because it's going to be interesting to see how the regulators react to whether or not a TCR, for instance, is going after a particular, let's call it mutation or antigen, is going to be agnostic to the tumor type. In other words, I'm going after an antigen A, and it's expressed in a lot of different tumors, and do I get a very broad label, or am I only restricted to tumor type Y that has antigen A? So I think that will be interesting, but once essentially the door is opened, it's replicative. Many, many TCRs will come through. Many, many of us have essentially built libraries of TCRs that can essentially go down that path. And then outside of the cell-based therapy, in oncology now, this is in my area, I think there the approval pathways will be building off the viruses. We've seen initially one with the oncolytic virus, and that I think is essentially going to be, again, biology that can turn the wheel to kick off other types of derivative processes where viruses are weaponized to have more and more to add essentially to perturb the tumor microenvironment, either as monotherapy or in combination with, for instance, other immunotherapies like checkpoints.
So we have a few minutes left, and Shelia, you touched on this, and Laurence, you touched on this as well, manufacturing and capacity, right? So most of the people in this room all have a need, and that need, okay, is to have manufacturing. And they touched on it on Oz's panel before that, to talk about, the decision to build internal, to use a CDMO, to try and find integrated solutions. So what's next? So when we think about the manufacturing capacities, that are out there right now, and all of us trying to get into the clinic, or trying to even push into later stage clinical trials, what do you see as some of the either biggest hurdles, both from regulatory standpoint or advantages that are going to go on as we look out into, 2020?
So just to start, and I'll go quickly here, Clearly, there are manufacturing solutions that are practiced in the local environment, particularly by academics, that look really interesting. The problem has been is when you try and scale those local, for instance, production of virus, lenti, retro, whatever, or production of cells, and then try and scale that to be a solution that can treat thousands and thousands of patients. It's non-linear. You can't extrapolate it. So I think that the next generation of manufacturing solutions will really have an eye to cost, because at the birth of this process, like 10 years ago, cost really didn't matter. It was based on a sort of the local academic environment. They were treating what he or she wanted in their local clinic. Now we have a responsibility to really look for cost solutions. So as new manufacturing ideas come on, they'll have to hit that filter. They have to hit that filter. It can't just be it works and therefore we will just grow it and kind of willy-nilly. It'll be essentially, there'll be a go, no-go decision on that. And then the second part that I think will solve the problem is that there's been basically in the world of manufacturing 2 types of people. The first type of people are sort of the gene jocks, fiddling with the genetic code. And the second types of people are the sort of the production people who say, okay, how can we get the genetically modified cell to do a lot of things and grow up to the right numbers?
You realize that all the quality people just cringed a little bit.
Yeah. No, but actually I think the solution is those 2 camps actually will really come together really beautifully. So in other words, production people will meet and say, look, I can't get this amount of virus made. And they'll go, for instance, to what you just did, and that is, okay, we have to go to a different genetic solution. It's not going to be a different packaging cell or a different, excipient that gets added to the process. It's going to be fundamental biology that will roll back right to the inception moment, right to the genetics. And the geneticists are not going to be in their own silo saying, look, I made a cell, now hand it off to the production people. There's going to be, I think, for successful companies, a real overlap now where those 2 essentially disciplines will unite to be a common manufacturing, scalable, cost-effective solution.
So real quick, for us, manufacturing is a multifaceted issue. It's not only your cell line and your plasmid, but it's also your vector. We often use our self-complementary vectors, which decrease the amount of virus required for therapeutic effect by about a log. Avexis uses that, the self-complementary vector in their SMA drug. And so that's one way, right? We reduce the manufacturing burden substantially just by using self-complementary vector. Also, if we improve the transduction efficiency of our vector, that increases the potency and therefore decreases the amount of virus required. The promoter, similarly, can increase the transduction efficiency, and so, again, that decreases the manufacturing burden. So it's a multifaceted approach that has to be thought about from the very beginning when you're designing your expression cassette.
So there is no one answer to this question because how many technologies are represented here. We, in our field, think about manufacturing simply as a 2x2 box of allo and auto and small and large dose. If you just simply, and you're just thinking about purely the cell, forget about the gene for a second, and which of those 4 boxes are you in? And that's a fundamental manufacturing difference, those 4 paradigms. So, and we, today, our first therapy is a cell, the authentic cell, but it's just the native cell. It's not engineered. And we chose to insource that. That is, we built a pilot GMP facility, and we're going to build a commercial GMP facility with the help of Bayer, we are wholly owned by Bayer now, with their help to build a commercial facility. And the reason we chose that is because we believe that that it is going to be so critical to control your own manufacturing destiny in this emerging field of pluripotent cell science. And we think, quite honestly, we're the most experienced people in the world at doing what we do, and the tech transfer burden is not worth the risk. So, so within just a pure native authentic cell, you're really think of it as one manufacturing process. We can do that today. We can make 5,000 to 10,000 doses at a time. We can But our next generation product, an example in partnership with our friends at Editas, where we're editing the genome of the pluripotent master cell bank, now you're incorporating all kinds of other components. You've obviously got guides and RNPs, but maybe you have AAVs or other elements. And obviously, now, are you going to try to manufacture all those components? So, I think the previous panel said it the same way I would, which is it depends on the piece of the puzzle. Our second generation products that will be edited will be sourcing multiple components. We do expect to be doing the editing itself in our own GMP facilities and the manufacture of the ultimate cell in our facilities. I don't see BlueRock in the next chapter outsourcing our manufacturing of the cell.
Yeah, I think I agree with everything that was set up here. I think the one thing I would just add is really interesting to start exploring ways of really dramatically enhancing the potency of some of these products. I think there's good, early preliminary data from clinical studies that even a small number of certain types of CAR T cells can have dramatic effects in patients. And so why do we need, a billion cells into a patient? If we can actually figure out the genetics and what sort of biological mechanisms enables a small number of cells to have a dramatic impact in the patient, then I think that's another way we can potentially try to grapple with some of these issues. And so really looking forward to, number one, looking at how some of the early stage clinical trials are playing out. And then I think we're going to see a lot of experimentation as to what's the right, allogeneic version of the cell to replicate the same effect, or certain tweaks we can make to the genome of these products to actually boost their activity, given some of those early stage data.
Okay, so we've got just a couple minutes left, so we'll close with one big thought. I'll kind of try to start us off. John Muir, so we started with talking about Muir Woods. You said that the power of imagination makes us infinite. And as we look forward, into the next decade about how we are trying to push gene and cell therapies forward, not just for stakeholders, but most importantly for our patients. When we think about that, it's how are we advancing the field? For me, I think one of the next things that's going to be critical, we're going to see more, is going to be combination dosing, where you're going to see multiple routes of administration for both, cell therapies or gene therapies or combination gene-modified cells. So I think that's going to be one of the next things that'll come up. We'll see hopefully more in 2020, especially going into this decade. So 1 minute each. Go for it.
Yeah, combinations is absolutely in the field of oncology. I think I'll go back to where we started, and that is you have to have scale. You have to be able to scale. The field is poised for success. So much money flows into this because at the end of the day, is immunotherapy going to replace chemotherapy? I mean, that's really our gift for our grandkids. That's where we're heading. Nobody wants to have pictures of kids or adults going through chemo anymore in our minds. To get to that point. Is one in which we have to have solutions based on cost and scale. Otherwise, this has the same type of biology or arc as bone marrow transplantation.
Yeah, I think we'll see a few things. Manufacturing will continue to be improved upon. Gene editing, I look forward to our friends at Editas and their data coming out with in vivo use of CRISPR. And I think there will also be a lot of interesting things about moving into pathway diseases. We have a lot of potential in AAV gene therapy, monogenetic diseases. We've made great impact, but I think there's a lot more that will be happening. And then finally, repeat administration. I look forward to working with our selective partners and our squeeze partners to see if we can do repeat administration.
I think I'm most excited about the intersection of the cell, the gene, and synthetic biology. If anything we've learned is the power of the engineered cell. We're engineering one element and we get this dramatic response. The cell has been evolving for 4 billion years. It takes in millions of analog inputs continually to produce the response. We've just scratched the surface of that. And so when you think about being able to engineer in logic gates and sense response switches and multiple component specificities where the cell has to in situ be activated multiple times in order to target the cell, if it's, for example, in immuno-oncology, it's profound. I mean, we are effectively, even though what we've done is amazing, we're children compared to what we're going to do when we've got full control of synthetic biology in an engineered cell, and that's a breakthrough.
Yeah, I share Emile's thoughts really deeply there. I think multifunctionality is going to be the wave of the future. We're starting to already see people bring products forward that have more than one function built into them. That's going to be really exciting. I think one thing I'll maybe just add, I think it's a little bit different in this room, is that the other really exciting thing just to build upon is, I was a professor at MIT before this. The amount of young talent that's going into this field is super exciting. I think the undergraduates, the graduate students who see the future of genetic engineering and synthetic biology and cell and gene therapy, I think people are inspired by the stories that you guys in the room here and this community has really enabled. And that's really enabling a huge influx of people, I would say, other than people who are chasing, AI and we're trying to work for Facebook right now. The next hottest thing in the universities is this area of bioengineering. And this is really just the beginning of it. We're just scratching the surface. I think this is going to be a really exciting decade going forward.
All right. Well, thank you for coming.